First speaker is Dr. Ping Wang. And Ping's scientific director at Janssen's R&D with the team and leads the company's efforts in the single-use qualification assessment of polymeric materials used in manufacturing, packaging, and delivery of large molecules. He's a board member of LC Consortium and an active member of BPOG and BPSA. Holds a BS and MS in chemistry from Beijing University, MBA from Drexel, and a PhD from pharmacy from the University of Georgia. Welcome, Ping. Good to see you again.
All right. Thank you very much, Michael. Let me share my screen first. Let me know if we can see that. Okay. Do you see my slide? I think.
Yep.
Perfect. Thank you very much again. Good morning, everyone. Thank you very much. Thank you, Michael, for the wonderful introduction. Again, I'm honored to be the first speaker to kick off this year's Pharma Ed E&L West Virtual Summit. The past 2 years, or at least past 20 plus months, has presented a unique challenge for our industry. This is not only for our pharmaceutical and the biotech industry, but also for our friends in the single-use system supplier industry as well. Personally, we have been, myself at least, have been working very closely with our procurement, tech ops, supplier quality, regulatory, planners, warehouse, and folks at many of other departments to come up with a strategy and strategies, actually, solutions to deal with our delays and the delivery of single-use systems and all the other shortages come along. Since the last time I discussed this topic here at this summit in June, situations are not getting any better, at least. Almost all the major suppliers are still telling us to be prepared for the longer lead time, which is absolutely true. And some even telling us to take our business to somewhere else because they cannot fulfill their orders. I have experience dealing with the suppliers over the past 10, 20 years. Never heard of suppliers saying things like that to us. And usually suppliers would try to everything they can to get our business and orders, not the other way around. I mean, the major cause obviously is due to COVID pandemic. And at this time of the period, and nothing is usual. That's why I'm here to share some of our experience of dealing with single-use system shortage. Also, we would like to get some feedback from our fellow colleagues in the bio industry and have some message to our suppliers as well. Here are some of the topics I'm going to cover in my presentation. First, a brief introduction about a single-use system shortage situation. Then we will talk about a little bit about dual source effort of single-use system because of shortages. And then dual source, triple source, and obviously, and then we will discuss assessment strategies of single-use system used in the COVID vaccine manufacturing process for both drug substances and drug products. We will follow up by some strategies for the long-term leachable study strategies for COVID vaccine drug product. And obviously, we are going to have some take-home messages to our suppliers. And finally, we will see what we have learned from the pandemic and this whole shortage situation. So now let's take a look of our current single-use system market situation here. I think probably at this time now, everyone is aware that this was true actually even before the pandemic. We have some shortages, and the demand for single-use system from bio and the pharmaceutical industry has been steadily increasing. And basically it's over the fixed facilities and it's still, this kind of fixed facility. Many companies, including us, are building all disposable facilities, not only just for R&D purpose or just early phase development, but also for the commercial production. And based on some of the surveys now, single-use system dominate over 85% of pre-commercial manufacturing facilities. This was based on pre-pandemic survey, so I think this could be even higher now. However, here is another caveat. I'm also hearing some of the biopharm industries companies are going backward, such as changing from single-use bioreactor to stainless steel bioreactors now. Again, this is not because they think the single-use, because stainless steel bioreactors are better or cheaper, simply because the shortages of single-use bioreactor, and they have to go back. So that is certainly is not the real good way to go. But obviously, I mean, this is a good news for our supply industry. For the suppliers of disposable materials, business is booming, and this certainly is the best sign to be in this business. At the same time, many suppliers, our partners, are doing expansion. We're talking from addition of clean room, assembly lines, or even brand new facilities, brand new sites. So, however, there are quite some challenges for suppliers, as well as for end users as well, to meet various quality, safety, and regulatory expectations. Again, how do I know this? Because we have been through many of the PAIs audited over the past 20 months and a couple of years in many of our facilities, our partners. As we know, we have drug vaccine manufacturing in 15, 16 different sites, all across global. We have been audited, inspected by many health agencies. So those are the key questions that we have been facing. Is big part of the auditor and inspection. So many of those inquiries are about single-use qualification and risk assessment. So now, let's talk about the shortages here. And again, let's say we already know that we have shortage problem. Besides the normal increase of demand for single-use system, even before the pandemic, this shortage was mainly caused by, we also have so many biopharma industry developing COVID vaccine in record time right now, right? We're talking about at least 3 major pharmaceutical companies, but there's a lot of other companies are doing similar thing as well. We're talking about billions of doses of vaccines. Suppliers just cannot keep it up. Again, let's ask ourselves, when was the last time we have made billions of doses of any other drug last time? I don't think that we have heard anything like that before, let alone in such a short time. Compared to the June when I first presented this about this one, at that time we didn't even have the booster doses approved yet. Now we have booster doses from Moderna, from Pfizer, from J&J. So you're talking about double that. And it seems to be this could very well be annual thing, moving forward. Who knows, right? So that is a big shortage problem here. So we are talking about billions of stoppers, or billions of vials and caps, along with unprecedented quantity of bags, filters, tubings, connectors, and assemblies. To manufacture them, we also need skilled workers, facilities, and space, raw materials, logistics to make those materials, and there are secondary, tertiary supplies as well. So along all with that, we also have to think about, we are talking about those are the parenteral drug product. We're talking about sterilization facilities such as gamma irradiation. Not sure if everyone noticed that we have a shortage of a cobalt-60 shortage, okay, and to cause gamma irradiation capacity shortage. So to overcome that, X-ray sterilization has become a hot topic now. So as a matter of fact, the BPSA is actively working with BARDA and other agencies to promote X-ray. So so let me just plug for BPSA. So if anyone wants to know more about this topic, you can contact James Hathcock, and James is going to be a speaker here today, and if not today, but tomorrow. So you can contact me or contact with James, and we can get back to the main topic. What is going on here? Okay. But let's get back to the main topic here. So, so the whole industry, it was just not prepared for this kind of shortage. The unprecedented demand surge caused industry-wide supply constraint. We are experiencing at least 2 to 5 times increase in lead time of purchasing orders without guaranteed delivery. We also have at least 9 to 14 months, which is about 4 times increase of lead time for some of single-use bags and filters. And the tubing delivery time is over a year now, which is 5 times the normal delivery time. So what are the business impact? So with a shortage, what are the impact? So Janssen is one of the COVID vaccine developers, and because we committed to make billions of doses of COVID vaccine, our need for very single-use system is tremendous, but the supplies are just not there. Therefore, for COVID vaccine related need, we can use U.S. government, it's called DPAS, Defense Priority and Allocation System status, to get ahead of with our other supplies, but the lead time is still unacceptable. Okay. And meanwhile, needs for all other projects are suffering even more because the suppliers did not even want to accept some of the order if they are not even COVID related. I mean, I'm not sure if this is still the case right now, but our other project will impact, that's for sure. Okay. Many companies started stockpiling at the same time or hoarding the materials to mitigate the risk of potential supply shortage. I mean, this certainly causes another, make the whole shortage even worse. There are also significant reliability risks for large molecule industry, both for short-term risks and long-term risks. We're talking short-term about 6 to 8 weeks stuck out, and long-term we're talking about 6 months plus strategic planning for the shortage. So the shortage and supply situation is very fluid and it changes daily. For example, confirmed order are unconfirmed sometimes, just days before the delivery date. So that is certainly cause a lot of issues for our planning and the manufacturing strategy. So what can we do? We have to dual source or multi-source, that's at least. And we have to do it fast because the shortage is imminent. As a matter of fact, half of my time over the past couple of years is spending on dual source effort. So some of our colleagues in the biopharma industry probably know this very well. In a normal pre-pandemic situation, if we want to qualify a second source of any major material, such as bagged filters for the manufacturing of later stage or the marketed product, it could easily take a couple of years to evaluation, testing, auditing, regulatory assessment, and so on and so forth. If we say that's the daunting job, that's not an overstatement. Fast forward to today, we have to dual source or multi-source hundreds of materials, and in a very short period of time, such as only a few months, and put them in production. It is simply overwhelming. We just cannot do it alone. We have to work together with the biopharma industry, with our suppliers, and with regulators as well, to make this really happen. So now let's take a look of our own situation. As you all know, we are developing the COVID vaccine to meet the medical needs and fulfill our commitment. We have to make billions of those things in a short period of time. Now we have to produce boost dose as well. Okay, so we have to engage as many partners as possibly can to make both drug substance and drug product. You probably already know this from the news. We have signed contracts with many of our partners, such as grants from Michigan and Catalan, we're both in Indiana and Italy, Aspen in South Africa, Rick Joffre in Spain, BioE in India, and Sanofi in France, and Merck both in Pennsylvania and North Carolina. And also we have IDT in Germany. And also we have quite a few of Janssen internal sites as well. So at least we have 15, 16 different sites to make drug manufacturing for the drug substance and drug product. So normally for the manufacturing of typical large molecules, we have total 2 or 3 sites, 1 or 2 sites for drug substance, upstream, downstream, right? And also sometimes upstream, downstream are in the same facility. And also we have another fill finish site, 2 or 3 sites at the most. But obviously this is not a typical case for COVID vaccine. We have 15 sites, and we have one major reason we can produce COVID vaccine in such a short period of time is we use so many different sites, and also we are using all disposable process, and or as much as we possibly can. By my last count altogether, we have at least 270, or at least high-risk single-use items. That's the last time I think I counted by at June, but I think this number certainly increased dramatically because dual source alternative source. So, but this doesn't even include so-called low-risk product contact materials such as gaskets, O-rings, connectors, and the probe. Those we consider as a relatively low risk, but we didn't even count on that. Those items usually have a smaller surface area or short contact time. Okay, in average, we have about 10 different high risk material for fill finish side, but 35 to 40 different items for drug substance. Of course, for the safety, quality, and regulatory purpose, we have to perform an assessment for every single high risk materials. For the EOA or BOA filings, we have to have independent sections for individual disposable assessment for each individual sites. Okay. So this is our normal disposable E&L risk assessment process. And since this is the E&L symposium, I only focus on the E&L part. We can certainly have. We also have to access other material quality attributes as well, but I'll just skip that. That's probably another topic for some other time. So normally each disposable material will be independently quantitatively assessed for drug product specific parameters and documented in a single report. As a filing strategy, a summary report will then be generated by summarizing all the quantitative details or individual material and reports provide as a drop-in report for the dossier filing. Okay, or for the PAI or regulatory inquiry. Each manufacturing site, we will have one summary report. Then drug product specific detail usually include how many material used, number of items used, and number of doses per batch, relevant extractable data and dosing regimen, and also E&L patient exposure will be calculated, and safety will be evaluated based on maximum exposure and PDE levels of individual extractables. So the advantage of this process is we will provide the details of every high-risk materials and a summary of regulatory filing inquiries. As a matter of fact, we are getting those kind of inquiries all the time. That's why we do it that way. As you can see, the drawback is the high workload to generate multiple reports. And I mean, this is usually manageable for a normal drug product development process and dossier prepare. We're talking 40, 50 different reports for one filing. And that is doable for the normal filing schedule and normal process. However, this is kind of difficult for our COVID because so many materials, so many sites for the EUA and the BLA filings. So this is a sample single-use system bound for drug product side. Let me give quite an example of a typical disposable bill of materials for fill finish side. I think this is a pretty generic for most of the large molecules. There are about 7 different stages. First, of course, we need to prepare the buffer solution. Then we will do a pooling and a dilution of drug solution. Then we will connect the drug solution through the various manifolds and transfer tubings to the filtration process. And then we'll do the bioburden reduction filtration of both buffer solutions and the drug substance filtration as well. And then we will follow by a sterile filtration. And then filling the compound in the sterile drug product into vials, and whether it's single dose or multi-dose. And the last column of this table actually shows the list of various disposable items for each stage. So I'm not going to the details of each material, but basically we are going to have various bags for buffer, drug solution, and the final filling. We also have bioburden and sterile filtrations, various TPE or silicone tubings and transfer manifold and connectors. All in all, we have at least 6 different kinds of tubings, not just 6 pieces of tubes, 7 different kinds of tubing in the process. 3 different kinds of bags, 1 or 2 different kinds of filters. Sometimes bioburden or sterile filtration could be the same filter. And also because of the pandemic, each of those materials will have to have alternative or even third options. So now let's take a look at the drug substance manufacturing product. Actually, I was trying to do a similar table like this previous one for drug product, but the process for drug substance is a lot more complicated, and the table would be too small to be useful to the audience. So I just put some summary here. In summary, there are about 12 different stages here, from pre-culture to cell expansion to virus production to clarification. We're talking about anion exchange, UFDF, the final fill and freeze of final bulk drug substance. As I mentioned, this could be a total disposal process. There are about 90 to 100 unique individual polymeric disposable in the process, and about 200 assembly throughout the process. Among those items, based on our assessment, at least 35 to 40 of them are considered as a high-risk item, and that requires the further E&L assessment. That means we would have to have 35 to 40 different reports for each drug substance site, and we will consolidate those into a single summary report as drop-in report for the dossier filings. So what are included in those drug product specific E&L risk assessment report? Obviously, we will have a summary section, right? Another report will have details about material tested, material used, and material number, when the catalog number, description of material, the size and length, and number of items used, and where the item are used. It will be contain a detailed extractable study and analytical assays such as extraction solvents, analytical assays. It should also have extractable study results, and such as identification and concentration of organic and elemental extractables, and the permitted daily exposure levels of those PDE of those extractables. It will have risk assessment details and methodologies such as calculated maximum patient exposure of all extractables versus the PDE levels, and further safety assessment if needed. It will also have margin of safety of those extractables and conclusion and remediation strategies if necessary. And the report also will include the reference of all relevant supporting documents such as guidelines, extractable reports, manufacturing details, DPFD and Pharma assessment, and source of safety assessment. Just want to add in is because sometimes we don't even pay too much attention about the references materials documents in the report, but we have been constantly asking by the regulatory agencies lately to where do you get this kind of assessment? Where did you get the PD? You know, how did you calculate the PD? And we would have that included in the summary report as well. Now, now let's review one more time and the single use for our COVID manufacturing process. We have, again, 15 different sites for drug product, drug substance. We have 270 plus different materials, and many of them, if not all of them, need alternatives. Just give you an example. Okay, we talk about, you know, Saint-Gobain C-Flex tubing is an industry standard. It's been used for the long time. It was in some kind of shortage years ago, you know, I think for quite a while now. But right now, I mean, this is really in real shortage. So we have to find alternatives such as, you know, Advanta Flex for Advanta Pure, which is a company from a great state of Pennsylvania. And this is everyone's alternative for quite a while. But so the lead time for Advanta Flex now has also dramatically extended too, because they are in such a high demand as well. So we are talking about people switching from C-Flex to Advanta Flex, now Advanta Flex back to, you know, C-Flex, and also there's other supplies coming along as well, such as for the medicinal, they have M-Flex, and this can go, at least can go on and on and on. So you can see this is actual number of disposal uses is much higher than 207. I mean, it is almost basically logistically impossible to prepare, you know, individual reports for every single disposal material in time for EOA and BOA filing. So what do? We have come up with some process. And the one solution we come up with is we only generate one summary report, all the material in one summary report, build a big table for that. This is a summary table for this is one big summary report. This table is actually a whole lot bigger than that, so longer and bigger. I'm just listing some of the key tabs in here and a few items here for illustration purpose. Okay, you will see the basic information of material, such as our internal material number, suppliers, description of item, material construction, and the catalog number. Then you will see the analytical results, such as extractables in the summary. We list only the most abundant extractable for the all relevant extractable solid. Okay, because if the most abundant extract is below the safety concern threshold or the PDE level, then all others will be too, right? So and then with the patient exposure level expressed in microgram per dose and the PDE levels of the extractables, our default PDE is based on the ICH M7 guidelines, unless our calculated maximum patient exposure level exceeds M7 threshold. So, and the last but not the least is margin of safety. So this is calculated by dividing the PDE by patient exposure level. As long as the margin of safety is larger than 1, we should be okay without going any further safety assessment. However, if for any reason, any of the margin of safety or any of the extract is less than 1, then our toxicology will go through further safety assessment. Okay, but again, we will still need to get all relevant information to generate reports like this. However, we just don't have to do all the individual reports. It's a significant saving of time. Obviously, I mean, there are pros and cons. Like I said, pros obviously is we save a lot of time, a lot of paperwork to do. And cons is we don't have all the details to answer regular questions. If we were doing the PAI or review process, we would still have to provide those kind of information day to day. Again, let's talk about the long-term leachable studies of the COVID. Okay. Again, normally we prefer to have at least 6 to 12 months of data for the BLA filings. Obviously, this is not possible for COVID. We don't. Our whole development process wasn't even that long, so that's impossible to get. So when we filed our EUA earlier, and we provide our plans for leachable studies along with our details of cleaning, the container closure system, such as the vials and stuff. We also provided justification based on our understanding and previous experience with those kind of components. So we have a plan, but we didn't have the data, anything yet. We have a protocol and a plans, stuff like that. We will need to do the study under the shelf life and accelerated conditions, such as 5 degrees C up to 24 months and 25 degrees C after 6 months. For COVID vaccine, we plan and experimental study are ongoing now. For 3 different conditions, 5, 25, and minus 20 degrees C. And we have flexible durations from anywhere from 12 to 24 months, depending on our shelf life conditions. So as you probably know, one advantage of J&J vaccine is it can be stored at 5 degrees C. We are also still exploring various shelf life conditions as we speak, and the shelf life studies are still ongoing as we speak. So here is what we are doing with the leachable study. We are using the first available multi-dose batch plus 2 additional multi-dose batches. So we have 3 batches going on with this study. We are going to use first batch for full shelf life from beginning to the end for every time point we are going to be testing all the way to the end of shelf life, whether it's 12 months or 24 months, depending on our real shelf life. And then second and third batch, we are going to for a beginning first time point and last time point. So this is to cover the possible drug product, a lot of variability. Meanwhile, we don't want to make this whole scope of Leachable study totally out of control. Here's another question. I just sometimes we try to going back and forth. We're still evaluating the options. When we are planning those 3 lots from the same facilities, because it will be possible or even necessary to use other combinations as additional batches to take care of a lot of variabilities, and possible their impact, different vials, different size, different doses. For example, can we use a single dose for second batch? We don't know yet. We are still evaluating this kind of options. So because that, we can tell you more details about in the next couple of slides. So this is the actual Leachable study planning we are going to do. Since the COVID vaccine drug product is so new, we don't really have long-term shelf life data yet for the risk. Leachable study, we are talking up to 36 months. This is what we are having now at 3 different storage conditions, minus 25 degrees C and 25 degrees C. I mean, our Leachable study probably would stop at a moment when we say, okay, our shelf life reach to, whatever time point we don't have to go, then we can start. But anyhow, we are planning for 36 months all the way to that. Bottom line is this whole planning is dynamic, is flexible, based on the data we have in terms of stability and shelf life. Here are some of the variables for final drug product. We have up to 9 different manufacturing sites, fill finish site. We have a single dose and multiple dose, right? And in terms of vials, we have regular type 1 vials, and also we have siliconized vials. Meanwhile, the vials could be coming from different manufacturers too, or from the same manufacturer, but from different manufacturing sites. You can see from this table, okay, the number of possible combination of final drug product in vials. It's a staggering, okay? So cost of testing all the combination, even a few of the combination would be astronomical. So it's almost impossible. We have to consider the cost. Okay, one study would cost at least somewhere from 100 to 200, depending on the combination, depending on your setup, depending on, in this particular case, we're using BSL 2-level lab, so that adding more complexity. So the cost is, times all the combinations. So that's just astronomical numbers. So therefore, we have to have use a risk-based approach, and we have to reduce the testing time and the cost. So what are the variables here? We're talking about in final drug products container closure system that may impact leachable profiles. And we could have a new primary packaging system, such as vials, stoppers, system. Could have changes to sterilization process of container closure systems, such as autoclave to gamma irradiation, or you may even have multiple sterilization process. We could have change of materials or construction. All of those highlighted here are considered as a major change. Meanwhile, we could also have some other minor changes too, such as change of locations, so CCS manufacturing site, fill finish side chain, or changing secondary packages. Now, how will those variables or change be impact our Leachable study approach? Actually, LC did an industry-wide survey on this very topic a couple of years ago back. So I'm going to give a plug for LC consortium here. So if you are from pharmaceutical or biotech industry company, and if you're not an LC member, and I really strongly recommend you, encourage you to join. Let me know if you are interested in that. So I have a website link and there, you can check it out. LC stands for Extractable and Leachable Safety Information Exchange. Here's a brief summary of approaches from the survey respondent regarding this major changes or variables. So majority of people will think we need to do the brand new shelf life Leachable study. We're talking about 74%. And 42% response that we would choose to do an accelerated study at a higher temperature. And some choose to do the extractable results comparison or simple paper assessment. Okay. And for minor changes of variables, a majority of survey respondents choose to do paper assessment only. Some choose to do extractable results comparisons. Some just go even further to do, smaller percentage go, full scale shelf life accelerated study on a case by case basis. Okay. So what should we do with our drug product situation with COVID? So far, we consider the variables in our situation are all the minor. So therefore, we will use only one configuration to shelf life and accelerated studies. We will perform paper assessment for all the other container closure system combination. Obviously, this is a science and a knowledge base and a risk base, of course. This is based on our understanding of container closure system material and historical data from our previous drug product. The risk-based assessment actually is in alignment with FDA's guideline. So what are some of the take-home messages from our suppliers? So I will say the last 20+ months was very challenging for all of us. Fortunately, we are getting out of this pandemic thanks to the vaccines mostly, and we are seeing a gleam of light at the end of the tunnel right now. So however, this does not mean the shortage of single-use system is over. On the contrary, I would say certainly not anytime soon. So we expect this to will go on beyond 2020 at least. Okay, we really have some message for our friend in the supply industry. We have seen the normal demand increase over the past decade, but 2020, 2021, and beyond 2022, the COVID pandemic also created great opportunities of supplies, especially some mid to small size players, okay, newcomers, and even some emerging market suppliers. In pre-pandemic era, those players would have a very tough time getting into the network of biopharm and biotech industries, especially in the large companies. Now they have a chance to compete with the big boys. So my message is for the major suppliers, and it's still very important, maybe even more important, to work closely with our end users and work and provide the best product. As I mentioned, it normally takes up a couple of years to evaluate and qualify a new major disposal material such as bags, filters. Now it's a few months. So this does not mean the standard for the single-use system disposal qualification is lower. As a matter of fact, on the contrary, based on our interaction with the agency during the PAI, the auditor, review process, the standard remains high, and it accounts for a large percentage of questions we receive for those kind of interaction with agencies. So for this supplier who could not provide sufficient and comprehensive data packages for evaluation right away, I would say it's probably challenging for them to get into the consideration for the supplier into our procurement network. However, on the other hand, I also have seen some newcomers. They're armed with comprehensive data packages, with BPOC data, we have cell sensitivity study data for the bags, we have USB EP testing data, and prepared to work with end user with us with all the answers and the capacity. So they will immediately get on the top of the list for the preferred vendors. So that is some kind of advantages and strategies and some of the new suppliers engage. So here's another message. So we need to provide as much info as possible for easy assessment and qualification. And especially, we only have a few months to consider that. The better the package, the easier to get it for the consideration. So this is going to benefit both end users like ourselves and the supplier. And a lot of people say, what kind of data we would need to have? I would say, let's start from the beginning. The BPOC electrical data, where is USB testing data? USB 87, 88, 665, 788, which is particles, and a EP 319 for the silicons, toxin, BSE, TSE, and for assembly, that'll be integrity, functionality. And the newer things will be nitrosamines, sterility. Sterility is not new, but nitrosamine is sterility. So for single-use bioreactors or bags for media preparation, meaning we need to have meaningful BDT, BBB levels and cell growth data to put it into consideration. Sometimes the true study data will be very helpful, and just simple extractable that may or may not be necessary to get into consideration because that is too important to miss. Last but not least, from a business continuity point of view, we have sufficient capacity and dual side for some of key components, very important to us as well. Now we have seen a lot of capacity expansion from our major suppliers. That is a very good sign. But on the other hand, I want to give another side of the story is, we are dual source a lot of materials from secondary, tertiary suppliers, And we also have seen some more material. We think everything looks good, qualify some material, put in the use. One example, we qualify a filter, all looks fine, paper, and same material construction. But when we put the use in filtration or some solutions, and solution turns to the different color. So that sort of is not what we wanted. And I'm telling the story is this is a whole lot more complexity in the process, even though sometimes everything looks good on paper, in reality, which may or may not work out. So that's something, would really help us, if supply can have, more in-use data to supporting that. That would be certainly very helpful. So let me to quickly go through this one. I'm not going to read it all through. This is a simplified checklist of disposal qualification process. We'll start with identification of need for disposal material, and this could be new application or could be a dual source. We will identify potential supplies, and this got a lot of technical and business evaluation. We'll evaluate the drawings, request prototype, and technical and quality evaluations. And then we will do, procurement, quality, inventory, SAP related activity, of course. Finally, we will issue, purchase order, close, the change control. This whole process is actually quite complicated, and you will so many different stakeholders from our end user side and our supplier side as well. So, this is just, a short list of things we are engaged. Again, so what I just want to briefly touch a few topics here. So the shortages due to pandemic assessment, strategy for large number of disposal in a short period of time, Leachable study impact, and finally messages to supply. So as bad as it has been, the pandemic also created a lot of innovations and opportunities for everyone, right? We have to develop new strategies to deal with the huge demand and challenges. We would like to work with suppliers for the speedy qualification and the new and alternative materials. And also we are hoping to see the increased capacity and the more updated data packages. And finally, thank you very much and enjoy the rest of the summit. And do feel free to connect me if you have any questions or comments. Okay, thank you very much.